field that's kind of in its infancy. There's a lot we really don't know about it. So I'm basically going to give, most of you have probably heard all of this before, but it's an overview and kind of a reminder of what epigenetics is and how it affects gene expression and disease outcomes. And if anyone has ever seen this book, it's a really good book. I read it, I don't know, maybe like 10 years ago. But it's really designed for the lay audience, but there's a lot of technical stuff that you guys could easily read through it. But it's just something, if you're interested in this topic, it was a really intriguing book. It kind of talks about the history of it, and what it means, and the direction the research is going. So does anyone want to define epigenetics? It's on the next slide, but I just need to see if anyone can take a crack at it. Yeah?
A way of passing down genes that doesn't involve changing the sequence.
Exactly. So it's gene expression changes that do not involve a change in the actual genotype. So it's a change in phenotype without a change in genotype. And there are basically two main mechanisms for this regulation. It's DNA methylation, which is a change at the DNA level, and then there's also histone modification, so there's changes at protein levels. And so one example of, when I was first exposed to this epigenetics field was actually in grad school. I did a rotation in a lab, and they were studying autism and schizophrenia in monozygotic twins. So these are identical twins who have the same genetic makeup, but completely different gene expression profiles in two different disease states. And so we were measuring basically changes in methylation patterns in the two twins. So it's kind of like a way, this is how this field has kind of emerged. So epigenetic changes can lead to gene expression changes. So this is the nucleosomes you can see here, and you have, when they're compact and tight, you have your genes inside. Your histones are made up, there's 4 histones in each nucleosome, and you can see the histones have methyl groups, and then the DNA is inaccessible when all of this chromatin is compacted. So the genes are not expressed. And you can see all of the histones have these little tails, and these tails are the ones that get modified to allow the chromatin to open and close. And so down here it's just demonstrating that we have these little histone tails. Right here they're showing acyl groups. Acyl groups bind to the histone tails to allow the chromatin to open up, the DNA is accessible, and then the gene can become active. So just like Dr. Llewellyn was talking yesterday about transcriptional control, when we have repressors bound and they hide the binding site for transcription factors, this is another way that you can close those sites so that the general transcription factors cannot reach the DNA. And so when it's open, it's accessible, those transcription factors can bind. When it's closed, the transcription factors do not have access to the promoters. So why do we care? Well, it's crucial to normal development and differentiation of different cell lineages. It can contribute to birth defects, degenerative and psychological disorders. There's a strong evidence for this in obesity and as well as cancer. And especially in cancer, this is like a huge field. I went on an interview for assistant professor position at a university, and I interviewed in 2 different departments, the cell biology department and biochemistry department. 9 out of 10 people I talked to in the department were studying epigenetics. So it's obviously a very important field in cancer. I do not right now study epigenetics, but it does explain a lot of the differences we see in the tumor microenvironment. So epigenetics is kind of a phenomenon that's regulated by the microenvironment. So the methyltransferases, which we'll talk about, all those things get modified by the tumor microenvironment. So it's really important in cancer, especially when we're looking at phenotypic differences in tumor cells. And we'll talk more about that when we get into cancer, but one of the reasons I'm putting this epigenetics lecture in early is so that you'll remember this later down the road. There's different ways of regulating tumor cell phenotypes. So exogenous environment can induce epigenetic changes to influence the gene expression, leading to the pathophenotype. So what kind of environmental contributors? Bacterial infections, drugs, alcohol, stress, all of these can affect histone modifications and or DNA methylation. So there are studies that can show that high-fat diet can lead to differential DNA methylation near lipid metabolism gene promoters. And then folate is important for methionine production, and so a folate deficiency can disrupt normal methylation patterns. Exercise induces methylation pattern changes in skeletal muscle and adipose tissue. So needless to say, these are all important processes that must be regulated, and so the epigenetic regulation is important to understand. So what is DNA methylation? So it's the covalent addition of a methyl group to DNA, mostly at the CpG sites. This modifies the gene expression. So the methyl group is added to the 5 carbon of the cytosine ring. So it's not 5 methyl groups, it's 1 methyl group added to the 5th carbon in the ring. And this is accomplished by DNA methyltransferases. And we'll talk more about those later, but there's several isoforms. There's a DNMT1, DNMT3A, DNMT3B. The DNMT1, I think, is for maintaining it, and the DNMT3A and B is for the initial methylation. So DNA methylation and gene expression. So we have a transcription factor that binds to the promoter. If the DNA is methylated, right, we cannot have transcription factor binding. So we have gene silencing. Also, we can have methylation, and which also blocks these binding domains. So one way is just prevent transcription factors from binding. The other thing it can do is methylation can attract these methyl CpG binding domain proteins. And what these do is they also prevent binding of transcription factors, but they can recruit other enzymes such as histone deacetylases and remodeling complexes to actually condense the chromosome. So there's basically 2 kind of outputs that can happen. Prevention of the binding as well as recruitment of other things that help prevent that from opening up again. So in the normal state, target gene is expressed. You have RNA pull binds to the transcription factor, and your promoter, your target gene is expressed. In the disease state, we have methylation of a promoter, and the target gene is not expressed because the transcription factor cannot bind. So can anyone give me an example of a gene in cancer that would satisfy this, that would cause this disease state?
Tumor suppressor gene?
Exactly. So if a tumor suppressor is suppressed and it's not being expressed, then it's not working, and so that can lead to cancer. You can have okay, so the opposite can also be true. So in the normal state, it's methylated. It's always silenced. The gene is not expressed. Transcription factor doesn't bind. In the disease state, we have the target gene gets activated by demethylation or hypomethylation, which is loss of methylation. And so we have RNA POL2 and transcription factors bind. That leads to the target gene being expressed. Can anyone tell me another cancer gene that would cause this disease state? Normally suppressed, but gets activated.
Cyclin D1 or
Yes, like an oncogene. So we'll talk a lot about oncogenes later, but these are genes that lead to uncontrolled proliferation and growth. And so those are normally suppressed in the normal state, but in cancer they get aberrantly activated and through an epigenetic modification is one way. Any questions about this so far? we're all good, genes are silenced, and we are fine. Okay, so DNA methylation patterns can be passed on after replication. So once the promoter is methylated by this DMTA3A or B, it tends to stay methylated under most conditions. And this is accomplished by DNMT1. So when DNA is being replicated, the methylation is, so the initial methylation is done by the DNMT3A and B, and then you see DNA replication, and the methylation is then copied onto the new strand by DNMT1. So it is heritable from cell to cell. So how do we evaluate DNA methylation? So there's a couple of different, there might even be more modern techniques. So one of them is to do sequencing of bisulfite converted DNA. So the way this works is you treat your DNA with sodium bisulfite, and that deamination will cause the carbon to change into an uracil. But if the cytosine is methylated, it's protected, so it is not deaminated. It maintains its cytosine. So when you do PCR amplification, you can see that obviously the U's become T's, but the C's stays C's. Another assay is, you guys have all probably heard of ELISAs. So these are enzyme-linked immunoabsorbent assays, and they're based on antibody binding. So in this case, you could immobilize your DNA on like a 96-well plate or whatever, and you can incubate it with an antibody that's specific to the methylated DNA, and then you can detect with a secondary antibody that has chemiluminescent reaction, and then you can just measure light. That's pretty easy. So that is the basics of DNA methylation. So next I'm going to talk a little bit about histone modification, so the second way of gene epigenetic regulation. And there are four kind of main avenues for this. It's methylation, acetylation, phosphorylation, and ubiquitination. And so the structural organization of eukaryotic chromosomes, you have the folded condensed chromatin, and then you have what's called, well, what looks like beads on a string, and so this is when it becomes more open. And so that's when you look at these electron microscope micrographs, you can see that it looks more like beads on a string here when it's open. And then when it's condensed, it looks, more tightly wound. And so that's just showing you what it looks like, your condensed versus your open nucleosome structure I mentioned before. Each of the nucleosomes has 4 different histones. There's 2 of each. Two of the 4, so there's 8 total. The H2A, H2B, H3, and H4. There's also histone H1, but all of these have these histone tails that can be modified in different ways. And that's shown here. So the tails have between 20 to 40 amino acid residues. And all of these tails are required for chromatin condensation. So they all get modified in order to condense the chromatin to prevent gene transcription. And this is just showing you a little bit of the histone modifications that can occur. So if you look at, so this is histone H2A. So you see there's a lot of acetylation. So this would be, what state, open or closed? Open, yeah. So this histone would have an open structure. H2B also, we see there's acetylation, there's phosphorylation, and ubiquitination. And then histone H3 in this example, you see there's methylation as well as acetylation. And H4 has a lot of acetylation, but some methylation. And so basically this leads to all of the combinations of these different acetylations and phosphorylations and methylations. It turns into this histone code, which is specific post-translational modification combinations in different regions, and these influence the chromatin function by creating or removing the binding proteins. And so a lot of this is due to charge differences. So it helps to enfold these. If you have a positively charged lysine, it reaches over and attaches to the negative charged DNA. So a lot of this has to do with charges of the amino acid. And so this table, do not memorize this table. It's just for your information, just to show you how you can have different acetylations that lead to activation. There's also phosphorylations that lead to activation, methylation that can lead to activation. Methylated lysines, most of them lead to repression, especially lysine 27 and 9. And then you can have activation and or repression on the ubiquitinated. So when you're reading journal articles, a lot of times you will see that they will mention specific histones. And so this is kind of like a resource for you if you're ever reading these or understand this one involved in activation or repression. So keep that in mind when you're reading papers and stuff. So this is just an example that shows that the actively transcribed genes, so the acetylated euchromatin, are susceptible to DNA's digestion. So in this example, erythroblasts can transcribe the globin gene, whereas these MSB cells cannot transcribe the globin gene. So when they add DNase, enzymes to cut the DNase, you can see that this would be an open chromatin because over time you can see that the DNA is being degraded, right? But this, in this case, is not being degraded when you add the enzyme, the DNase enzyme. There is no degradation because we know it's condensed, okay? So this would be one way to see whether your chromatin is condensed or not around a certain gene. So heterochromatin, again, inactive, condensed. You have methylation in certain lysines, 9 and 27. Those are the main ones that are leading to kind of condensed close structure. The active euchromatin is marked by all these acetylations. And you can see, like within the nucleus, there are several chromosomes that are open, several chromosomes that are condensed. And so if the repressive histone marks provide an advantage, the survival advantage for the cell, so the histone will recruit further suppressive proteins like deacetylases, methyltransferases. And methyltransferases recruited to the histone can reach over and methylate nearby DNA. This is how this all kind of stays together. Okay, any questions about that? Good. Okay, so this is just more of a slide to summarize sort of the different enzymes that are involved in these processes. Histone acetyltransferases for transcriptional activation. These transiently bind to DNA histone acetylase complexes for regional acetylation. The histone deacetylations are necessary for repression of genes. DNA bound repressors they transiently bind. So a deacetylase is taking off the acetyl group. Histone methyltransferases, they're necessary for the heterochromatin. And functions in co-repressor complexes to repress transcription through the methylation patterns. Histone lysine demethylases catalyze removal of the methyl groups. So the histone modifications are fairly transient and rapid and easily changeable, whereas the turnover of the methyl groups and especially DNA methylation is more stable, so it's harder to change that. So the histone modifications are much easier to change. It's good if you're a cancer cell and you want to be very plastic and change your phenotype a lot more. Okay, so what would be a possible consequence of increased activity of a methyltransferase near a tumor suppressor gene? too easy, isn't it? Go ahead.
You're at risk for like cancer or that cell becoming cancerous.
Yeah, exactly, because if you're suppressing, the example earlier, if you're suppressing a tumor suppressor, that's gonna lead to growth, okay? All right, what about increased activity of a demethylase near pluripotency genes on differentiated cells? So differentiated cells are not pluripotent, right? They have a specific function. They're specialized. So what if you had a demethylase? Demethylation, taking off the methyl groups.
They start expressing genes that they're not supposed to.
Yes. And they can start making cell types that they're not supposed to, right? So these are kinds of questions that would be on the exam, okay, just to be aware. So you want to kind of think about, if you're given this enzyme and you know what it does, what would be the outcome in the cell? What would be a potential caveat of using inhibitors of methyltransferases or demethylases to treat cancer? Given what I've gone over today, you guys think of a possible.
They're not specific, so you can hit other targets that you don't want to.
Yeah,
It's
so
not
we're
mess
gonna-
with other processes.
Yeah, so we're gonna turn off that oncogene and then turn on the oncogene, but then turn on other oncogenes, right? So that's one of the struggle, I think one of the challenges of this field is finding very specific inhibitors of these different methylases at specific times. And so that's a challenge in the field. I mean, it's a challenge in, there's a lot of challenges in the cancer field. That's one of them, one major one. So it's such an interesting phenomenon to understand how these changes can happen, but they're so broad, and these are mechanisms are all throughout the genome, so you can't just turn one off and put this one on. So what people do are, can anyone think of another way that you could modify a treatment to combat this problem? I'm leaning more towards the cancer people. You can think of a way. This is not a question that would be on the test, but maybe on the final. After we get the cancer stuff. So sometimes there are specific pathways active in cancer cells that can turn on these enzymes. So if you can inhibit a particular pathway that's only active in the cancer cells that can turn off these things that are happening in those cancer cells, that would be one way to kind of overcome it. Okay, so it would be like this extra methylation is happening in this tumor cell because of this pathway that's being turned on. And it can also be kind of like a tumor microenvironment issue if you targeted whatever in the tumor microenvironment is causing the activation of that enzyme. So it has to be something that's specific to the tumor cells. Okay, so how is methylation maintained during replication? Basically, there are histone methyltransferases, and they bind. So when you have replication, the parent strand maintains some of the methylation, and then these histone methyltransferases will bind and then methylate the neighboring histone. And so you end up with the same that you started with, the same amount of methylation that you started with. So it's inhibited. And then, so I mentioned before, methylation is required for the condensation of heterochromatin. So there are proteins that have chromodomains, and chromodomains are just domains that bind to methylated histone tails. And so these can also bind other chromodomains and recruit histone methyltransferases for spreading of the heterochromatin. And so that's kind of shown here. So you have these methylated histones, you have binding of this chromodomain protein HP1 to the methyl group, and then that will lead to recruitment of more HP1, and it also can So it can bind, it can methylate H1, it can methylate next door, and it can also recruit more. And so that's how you kind of have this condensed chromatin. And this will continue until it reaches, so it will continue throughout the chromatin until it reaches this sort of boundary element. And that boundary element is just a protein that's a non-histone protein, and it basically shuts down the process. And a similar mechanism happens for the euchromatin, and in this case, instead of the chromodomines, we have proteins with bromodomines, and those bind to acylated histone tails, and then you have a similar spreading of the acylation. Okay. So one of the most important functions of epigenetics, or an epigenetic mechanism in methylation, is X chromosome inactivation. So females have one of their X chromosomes are inactivated. And so we have this random inactivation of either X chromosome in every cell. And this is characterized by hypoacetylation and hypermethylated histones. So we have less acetylation, more methylation. And it's controlled by Exist, and this is a long non-coding RNA that's actively transcribed on the inactive chromosome. So on the active chromosome, we have this T6 gene that's transcribed. And what do you think T6 probably recruits? It's active. Okay, so T6 is on the active chromosome. So this is the chromosome that's active. So that's going to recruit things like acetyltransferase, things that's going to keep the chromatin open. The inactive chromosome has this long non-coding RNA Exist. And what this does is recruits more methyltransferases, and it can actually spread along the chromosome, and I think I have that on the next. So you have this long non-coding RNA, and it spreads along the inactive X chromosome. It recruits these binding proteins, it recruits methylating proteins, and then basically you have all of, so the purpose of this slide is just to show you that it's not just a long non-coding RNA that recruits methyl. There's a lot of proteins involved in this process. But it starts with this long non-coding RNA that kind of gets transcribed and binds to different sites along that same X chromosome. And it leads to, again, recruitment of methyltransferases at the inactive X chromosome. So this is like one of the most important mechanisms of epigenetics. And there's a lot of, so this whole, so this is like a normal thing that happens, but sometimes whole chromosomes will get silenced, and we have, they're not supposed to get silenced, and that's when we have diseases. Okay, tools to measure you guys probably already know all these. So tools to measure post-translational modifications. So of course you can do Western blots. You can do immunoprecipitation, mass spectrometry, and then you can also do enzyme assays. So these enzyme assays, they measure byproducts of the enzymes, such as this adenylsulmethylline. Deamethylase activity leads to formaldehyde byproduct. Acetyltransferase leads to NADH byproduct. So you can measure basically the activity of these different chromatin modifying enzymes. And of course your chromatin, is everyone familiar with immunoprecipitation, how that works? So you basically have an antibody that can recognize a specific protein, and then you can use antibodies to pull that out, and then you can run it on a gel, or you can look at what's bound to that. Okay, so if you have your chromatin, so you have all your histones and your DNA, and then you add an antibody that's specific to a specific modification, you can pull that out, and you can basically sequence the DNA, and you can do qPCR to see where it's bound. So you can look at where methylated histones are bound, what genes are bound.
Are the antibodies like that are recognizing these things? Are they like created thinking that they'll recognize that area, or is it just kind of like experiments have shown that this recognizes that, and we just buy it from a
I think
place?
they're created to recognize it.
interesting.
They're not, I don't think they're naturally
If
occurring.
your body made a natural antibody that then you have like a huge
Yeah.
autoimmune response.
Uh,
So we make them, like mice and rabbits.
Okay.
We make them specific to recognize those sites.
So I forgot to talk about ChipSeq yesterday. I guess I didn't realize there were 2 more slides. So I'll talk about that on Tuesday.
Okay, so, there are, despite the, caveats, there are some epigenetic therapies out there. So there are some histone deacetylase inhibitors that are in FDA approval, and they're probably in clinical trials or were in clinical trials. There's inhibitors of methyltransferases, and they inhibit specific, specific recruited proteins. So if you have an overexpression of something that leads to methylation, you can inhibit that. There's also DNA methylating agents that can also have a secondary effect. So this is basically if you have a DNA methyltransferase, you can induce genetic instability by adding chemotherapy. That leads to a damage signal that leads to apoptosis. There's lots of different ways you can try to use epigenetic drugs to target cancer or whatever therapy, whatever disease you're trying to overcome. And I had one more slide, but I'm not gonna go over this. It's just another regulation of it. So does anyone have questions about epigenetics? Most of this should be a review for all of you, but good reminder, maybe, I don't know. Questions? Everything is crystal clear. Then I would get emails later, what about this, what about that? Okay, well, so this is, again, just a basic intro, so expect questions like I posed in there in the lecture on quizzes and exams and stuff. Okay. So there's like, I don't know, 15 minutes left if the groups want to meet. So the groups were posted, did everyone see that? I know who groups are, and I posted papers.
I did too.
Did we unhide the weeks?
I didn't hit mine. I wasn't sure if you
Oh.
posted yours.
All right, I'll unhide the weeks when I get back to my office so you can access